Chapter 8 covers metabolic pathways, and the big chunk of this chapter is the reactions necessary for cellular respiration. Now, I mentioned in the notes that I don't really love the pictures that come in your textbook, so I pulled these pictures from a different book. There are lots of different ways to study metabolic pathways. You should have had these before, at least in a simplified form in previous courses. If you didn't, you're going to have a lot of catching up to do. One of the things I think that I mentioned in the notes is that I was not required to learn enzymes when I was at the sophomore level, so I'm not going to make you learn the enzymes necessary for each individual step, but you do need to know the intermediates that occur within the pathway, and this picture shows you that. So this is the glycolysis pathway. It starts with a molecule of glucose. This is only showing you the 6-carbon chain. It's not showing you the oxygen and hydrogens, so keep that in mind kind of as we go through there. It will throw in phosphates as these little yellow balls. Those are going to be very important for us. So the first step that happens is we spend an ATP to phosphorylate glucose into glucose 6-phosphate. So something from chemistry that I'm hoping that you remember is the number tells you which carbon the phosphate is on. So honestly, this molecule should be flipped, but whatever. So this phosphate is on the 6th carbon, which is what makes it glucose 6-phosphate. Then remember that fructose is just an isomer of glucose, so we just do a chemical rearrangement and this becomes fructose 6-phosphate. From there, we spend another ATP to phosphorylate the fructose 6-phosphate to make it fructose 1,6-biphosphate. Notice that now we have a phosphate on the 1st carbon and the 6th carbon. This molecule splits into two 3-carbon molecules called dihydroxyacetone phosphate. Now, this molecule is very unstable, and so it pretty quickly changes into glyceraldehyde 3-phosphate or G3P. This molecule is also going to play a role in photosynthesis later. It's an important little molecule in a lot of different places. Here's where we start to strip electrons off, and so we reduce our first electron carrier from NAD+ to NADH. We do an addition of a phosphate group in here to make this 1,3-biphosphoglycerate. From there, we're going to strip a phosphate off. That's going to help us with some substrate level phosphorylation so we can convert ADP into ATP. That also turns our 1,3-biphosphoglycerate into just 3-phosphoglycerate. We lost that first phosphate up there. From there, a chemical rearrangement, we move the phosphate over to the 2nd carbon so it becomes 2-phosphoglycerate. We strip off a water molecule and make it phosphoenol pyruvate or PEP. And then, oops, I didn't mean to do that, sorry. And then PEP, we're going to do another substrate level phosphorylation, take the phosphate off, add it to ADP to make ATP, and that turns this into pyruvate. Notice that all of these things from the dihydroxyacetone phosphate all the way down to the pyruvate, we're doing that 2 times because we split our 6-carbon molecule into a 3-carbon molecule. Everything that's up here in the gray boxes are what are known as intermediate metabolites. That means these molecules can either proceed through glycolysis, or if the cell needs them to do something else, we can filter them off and then use them in other pathways as well. And so that's why those are special and in those nice little gray boxes. And so that's what's happening during glycolysis. One other thing to keep in the background of your mind is we need separate enzymes to carry out every one of those reactions as we go through there. So remember, enzymes are important. They are genetically based, so organisms have to have the genes to make the enzymes to carry out all of these steps right here. Okay, from there, we're going to enter into the transition step and the citric acid cycle, which I was taught the Krebs cycle. Your book still calls it the Krebs cycle. The transition step starts with pyruvate. It strips a carbon off, and so the pyruvate becomes an acetyl group. Coenzyme A is added to the acetyl group to make acetyl coenzyme A, and then acetyl-CoA can be combined with oxaloacetate to become citrate. This is where we get into the actual cycle part. All of the rest of that was the transition step up here. Now, once we have citrate, the citrate is going to do a chemical rearrangement to become isocitrate. Again, it's just an isomer of citrate. From there, we strip some electrons off to reduce our NAD+ into NADH. That's going to create alpha-ketoglutarate. We're going to strip some more electrons off. The alpha-ketoglutarate is going to become succinyl coenzyme A, and we're also going to reduce NAD+ to NADH. The succinyl coenzyme A is going to get converted to succinate, and as we do that, we're going to do a substrate level phosphorylation of usually ADP into ATP. From there, we're going to strip some electrons off of succinate to make it fumarate. Those electrons that we strip off are going to help us reduce FAD to FADH2. Fumarate has a water added to become malate. We strip some electrons off of malate to reduce NAD plus into NADH, and that helps us also recreate the oxaloacetate that helped us start this whole entire cycle through here. Now, in the notes or in the PowerPoint, I don't remember where I put it, there is a stupid mnemonic that I have remembered forever to help me remember the order in which these things occur, and that is, can I keep selling sex for money, officer? So if you can remember the stupid mnemonic, it can really help you remember what's going on during the Krebs cycle off down there. Now, the electron carriers that were generated during glycolysis and during the Krebs cycle, that's our NADH and our FADH2, those are going to be critically important for oxidative phosphorylation, which this was in my days just called electron transport. But electron transport is necessary to help set up chemiosmosis, which then can help us set up our oxidative phosphorylation. So all of these things sort of merge together in the most ATP generative pathway that cells are going to have. Both the Krebs cycle and this oxidative phosphorylation in eukaryotes is going to happen in mitochondria. In prokaryotes, remember, they don't have mitochondria, and so they're just going to be doing this on their cytoplasmic membrane. The membrane is critically important because we have to have these proteins embedded in a membrane. Those proteins are going to shuttle electrons from one to the next, which is why it's called an electron transport chain. As they do that electron shuttling, they're also going to pump hydrogen ions across the membrane, either the inner mitochondrial membrane for eukaryotes, or just the cytoplasmic membrane for prokaryotes. That is going to set up this huge hydrogen ion concentration gradient where there's a bunch of hydrogen ions on one side of the membrane and very few on the other. Those hydrogen ions create what is known as proton motive force, and then those hydrogen ions can flow, following their diffusion gradient, through a protein called ATP synthase, which is a literal little turbine in cells. As the hydrogen ions flow through, they spin this turbine, and then that spinning force helps the protein to add a phosphate onto ADP to make ATP. This is the part that makes this oxidative phosphorylation. The oxygen is the terminal electron acceptor in the electron transport chain, oxidative. The phosphate getting added off over here, that's our phosphorylation. So that's why all of this is called oxidative phosphorylation. Now, coming back to uniqueness in different organisms, there's different proteins used in different organisms, different species. The picture in your book is showing cytochrome C. Some organisms have that, some don't. Prokaryotic. Sorry, I'm going to pause because my puppy's excited. Okay, so I think I had ended with different organisms use different proteins. This cytochrome C oxidase right here, it's special because we can use that as a test since some organisms have it and some don't. You guys are actually going to perform the oxidase test in lab. It's testing for that little protein that's part of the electron transport chain in some organisms, but not all. The electron transport chain that I want you guys to learn the running parts on are these right here. This is again a picture from another book because your book doesn't really have a good picture of this, but this one is a fairly simple one. It's also fairly common amongst prokaryotes. And so here we go. NADH dehydrogenase is this first yellow protein right here. It accepts hydrogen ions and electrons from NADH. So this strips the electrons from our reduced NADH, converting it back and oxidizing it to NAD+. Those electrons are going to get passed to ubiquinone, which some instructors have called it ubiquinone. I don't really care which way you want to say that. It also uses that shuttling of electrons to help it pump hydrogen ions across the membrane. Remember, this is the cytoplasmic membrane because this is the prokaryotic electron transport chain. Succinate dehydrogenase is going to be accepting electrons from FADH2. It's going to pass those electrons also to ubiquinone. So ubiquinone is accepting electrons from both NADH dehydrogenase and succinate dehydrogenase, but only the NADH dehydrogenase is going to be pumping hydrogen ions across the membrane. Ubiquinone is going to shuttle the electrons over to ubiquinol oxidase, and it's going to give the electrons usually to the terminal electron acceptor, which is going to be oxygen. That converts the oxygen into water molecules. As it does that, it's also going to be pumping hydrogen ions across the membrane. So these two proteins here, not only are they part of the electron transport chain that are transporting electrons, they also help to set up that proton motive force by pumping hydrogen ions to the outside of the membrane. That proton motive force is going to help, again, drive ATP synthase into the spinning turbine action and then phosphorylate ADP to create ATP. So this is where we're actually generating our ATP right here. Once again, some organisms have different protein carriers or electron carriers, pardon me, than this, but this is the one that I want you to learn. All right. From there, fermentation. A bunch of different organisms can carry out different types of fermentation, and a lot of these are biochemical tests that we end up using in labs to help us identify unknown organisms. The example that I'm going to give you here, I'm going to do two. So this 2,3-butanediol right here, the VP test and the MRVP, the Voges-Proskauer, it detects this 2,3-butanediol. The mixed acid tests, that's the methyl red test, which is the MR part of the MRVP. So only organisms that can ferment to produce these products will pop positive on either the MR or the VP part of those tests. I am also showing you some foods or drugs that are sort of related to those things, but these seem like they're really esoteric, but they're very important in a lab. I'm going to pause again for weird puppy time. All right, hopefully that tired him out long enough for me to finish this video. This slide I'm really only leaving up here because I like this picture in terms of how it explains how we can use other molecules to help us fuel metabolism. So this is showing you glycolysis, the transition step, the Krebs cycle, and then oxidative phosphorylation or electron transport down here at the bottom. It shows you that your fats have to get broken down to glycerol and fatty acids. That glycerol can be converted into a glyceraldehyde 3-phosphate and fed in during glycolysis after we have split our 6-carbon into 2 3-carbon molecules. The fatty acids go through beta oxidative. Don't bite me. Calm down. Sorry. Never try to record videos and have a puppy at the same time. It's ridiculous. Okay, the fatty acids are going to undergo beta oxidation, which is where we snip off 2 carbon units from the long fatty acid chain. Those 2 carbon units are acetyl groups, so we can feed those in as the acetyl group, which can be combined with coenzyme A in the transition step to make acetyl-CoA, which will again be what combines with oxaloacetate to start our citric acid cycle. The proteins get digested down to amino acids, which have to be deaminated because remember, there's no room for nitrogen in any of this pathway. Once the nitrogen's removed, it really depends on which of the 20 amino acids you have, where it's going to get fed in, and I am not going to make you memorize which specific amino acid gets fed into which part. I really just want you to know they can get fed in either in glycolysis or the transition step or the Krebs cycle, and as long as you know that, then I'm pretty much happy. Okay. Your super brief rundown of photosynthesis. Regular old photosynthesis, like what plants do and what cyanobacteria can do, has 2 different sets of reactions that occur. What I was taught as the light reactions and the dark reactions, what are now called the light-dependent reactions because they are dependent upon light to work, and the light-independent reactions because they do not need light to work, but light can be present and they can still go on. Okay. The light reactions is where we're going to be generating the energy necessary to carry out the dark reactions. So really what happens during the light reactions is light helps power another electron transport chain that's going to help us make ATP, pardon me, down here, and then some NADPH, which is another electron carrier that really only gets used in biosynthesis. It's related to NADH, but it's not the same thing. These 2 things then get fed into the dark reactions and help us power the reduction of carbon dioxide into an organic molecule like glucose or sucrose or whatever. And then this, as it uses up the ATP and the NADPH, it creates ADP and NADP+, which can be regenerated over here. So both of these 2 reactions are necessary for the other to occur. Now coming back into a little bit more detail on the light-dependent reactions. Again, it's very, very similar to electron transport as it happened in cellular respiration. The difference is we're not using electron carriers to bring electrons to the electron transport chain. Instead, we're stripping electrons from some other molecule. What molecule we strip it from depends on whether we're doing oxygenic or anoxygenic. For oxygenic, we're stripping our electrons from water. Once we strip the electrons from water, we bounce those electrons up to a high energy state and then let them run down an electron transport chain to do two things: to help us reduce NADP plus to NADH, and to help us set up proton motive force so that we can use ATP synthase, the exact same ATP synthase from earlier, to generate more ATP. So that's what's happening off over here. Again, very, very similar. It's just that our electrons come from different things, but aside from that, we're really doing the same stuff there. I hope that helps you understand some of this. And again, the more you repeat it, the more sense it will make. And I really hope this helps.